Welcome to Microbial Growth class. Today we are going to discuss the respective parameters used by microorganisms to grow and develop. And usually these microorganisms grow through reproduction, or what we call multiplication of their cells. They can do that through sexual reproduction and asexual reproduction. Bacterial sexual reproduction involves the direct division of either bacterial cell or either microorganism cell into 2, 4, and 8 respectively. However, from here you can see that binary fusion starts with the condensation of the chromosomes, division of the organelles within the cytoplasm, and then the division of the cell wall and the cytoplasm membrane to give 2 daughter cells. Another form of asexual microbial reproduction is called the budding. The budding involves the elongation and condensation of the chromosomes at the first stage, then subsequently division along the centromere and elongation of the cytoplasm membrane together with the cell wall to produce. Another form of asexual microbial reproduction, most especially the fungi, is by the production of the conidia. The conidia is an extension or detachment from the conidial spore produced from the extension of the hyphae. The hyphae is the fibrous part of the fungi which extend to produce conidial spore, and by division they produce daughter conidia. These daughter conidia, when detached, they can maturate within some certain period and eventually produce other set of conidial spore from the hyphae. Another form of asexual reproduction is called the cyst formation. This is undertaken by both bacteria and protozoans. The first stage starts by the condensation of the chromosomes, multiplication of the chromosomes to an encystment of the respective chromosomes to produce several cysts. These cysts, in turn, after they finish enclosing within the cyst, the cell open and it produces daughter cells and are released into the environment. Example of these organisms that partake in cyst formation as a means of multiplication include Azobacter agilis, Azobacter tropicalis, then protozoans, Giardia lamblia, Entomoeba histolytica, Cryptosporidium favum. The last form of asexual reproduction is by formation of the endospore. Formation of endospore usually takes place in bacteria, and it first involves the arrangement and condensation of the DNA. The DNA, after the condensation, separate into 2 along the centromere and migrate to the symmetrical end of the cell. In the symmetrical end of the cell, a line of symmetry is drawn, and the line separated the cell from the first chromosomes that were parted away. After that, the endospore is formed, dividing the cell into 2 within the cytoplasm, and subsequently the cell divided into 2 to produce 2 cytoplasm, 2 chromosomes, which give rise to 2 daughter bacteria. This category of bacteria that undergo endospore formation includes Bacillus cereus, Bacillus anthracis, Bacillus thuringiensis, Clostridium botulinum, and Clostridium tetani. The second category of microbial reproduction, which leads to multiplication and development of this microorganism, is sexual reproduction. Sexual reproduction has three divisions: the conjugation, transformation, and transduction. Conjugation involves the transfer of independent DNA, which is called the plasmid, from the donor bacteria to recipient bacteria through a channel. This channel is called the pili. Pili is made of a protein called the pilin, and the plasmid carries a combination of genes that encode a transfer by the production of relaxosomes and transperosomes. After the transfer, the plasmid incorporates itself into the DNA of the recipient organism and can mutate or convert the bacteria into a pathogenic or virulent organism, or it can convert the bacteria from normal to resistant bacteria, which in turn make it very pathogenic. An example of this kind of bacteria that conduct conjugation include Mycobacterium smegmatis and Escherichia coli. Sexual reproduction is an equivalent of sexual reproduction in advanced organisms in bacteria. It includes conjugation. Conjugation is usually the transfer of plasmid and independent DNA from donor bacteria to the recipient bacteria. These bacteria attack the F plus bacteria and the F negative bacteria. The plasmid encodes the transfer of the DNA using transferome and relaxosomes. They transfer the plasmids via the pili, and after the transfer, two daughter bacteria were produced. These bacteria that can do such kind of thing include Mycobacterium species and Escherichia coli, specifically Mycobacterium smegmatis. The second form of bacterial sexual reproduction is called the transformation. Transformation involves the interchange of the bacteria through the abduction of independent plasmid within its environment. This usually takes place during culture. However, the bacteria have to be conformative to the recipient or donor bacteria. That's why only some selected bacteria were able to conduct the transformation. They include the Streptococcus pneumonia and Escherichia coli. The independent DNA usually finds its way to incorporate itself into the recipient DNA. This in turn causes mutation, and the mutation causes a change of behavior for the bacteria. Maybe it enhances its virulence and resistance. The last category of microbial sexual reproduction is transduction. Transduction involves the infestation of tipo patch into the Escherichia coli bacteria. They start that through penetration, attachment, then penetration via the patch spikes. This follows the injection of the DNA. The DNA in turn hijacks the replication mechanism of the bacterial DNA. It's encoded and incorporated DNA into the bacterial chromosomes, therefore produce the patch particles. The patch particle can utilize either the lytic or the lysogenic phase. In the lytic phase, they burst through the Escherichia coli cell and infest or attack other immediate bacterial cells, or in turn stay dormant via the lysogenic phase. This involves the Take a recess and answer the following question. Define the microbial reproduction. Please find microbial sexual reproduction and explain conjugation. Thank you for listening. Go through it and subscribe to my channel. Thank you for listening. Go through it and subscribe to my channel.